In this screencast, we're going to talk about protein synthesis, the last step in the processes of going from a DNA code all the way to a finished product of gene expression. Once again, this is a review of a picture that you've seen a number of times. This shows where the DNA is in the nucleus and where the proteins are actually put together out in the cytoplasm at the ribosome site. So remember that a gene is a specific base sequence along a DNA molecule. So just picking some points, but all of the bases, and there can be maybe a few hundred to thousands of these bases would make up a particular gene. A gene is eventually going to code for a specific trait. That trait might be eye color, hair color, all the ones we've listed and talked and discussed about over a number of times. But a gene is a sequence of DNA, and it's a specific sequence of bases that is going to code for a trait. So a sequence of DNA, a specific sequence of bases on a DNA molecule, is called a gene. And that gene is going to code for a protein, a specific protein that is then going to be used in the expression of traits. So translation is the process of taking amino acids, building proteins for gene expression, or for the expression of traits in a living thing. Translation is the process of building a protein molecule. So you've seen examples of this, but proteins are the finished product of translation. But we still think of protein as just some imaginary or hypothetical molecule. You know that it might be a component of some foods, but proteins are the structural components and the chemicals that run chemical processes in bodies. So proteins are everything from the pigments in eyes and skin color to the structures that actually build the muscles and muscles in your body. They are hormones that are released by glands in the body, such as adrenaline, testosterone, estrogen. They are enzymes in the body that help you to do things such as break down lactose products, milk products. Lactase is an enzyme that does that. The musculature that builds your tongue in such a way that you can or cannot roll your tongue. Protein's a huge class of molecules that do structural things and do functional things in the body. So for this slide, we've really narrowed it down to 6 basic steps of translation. Again, translation is where you are going to go from the mRNA sequence, the sequence of bases on the mRNA strand, to building a growing protein chain. You can stop this, pause on this screen, read through these for yourself, review it again and again and again. You can look up YouTube videos that will explain it probably better than I do, but take the time just to understand the step-by-step process. Not to memorize it, but to get in your mind a scene where you're going to go from the DNA sequence of bases to building an mRNA strand, and how that equates or ends up with a product of amino acids strung together to form a protein molecule. As you go through and look at this process, just remember that at the starting point, the DNA molecule that makes us who we are and makes us unique, there is virtually nothing random about the order of bases in that molecule. Therefore, there's nothing random, or in other words, it's very specific as to the order of bases in the mRNA sequence. If that order of bases is specific on the mRNA strand, then the order of anticodons, or the order of tRNA molecules that come into play, is going to be specific. Therefore, in turn, the order of the amino acids is going to be specific. And finally, the end product, which is the completed protein, is going to be specific and unique for all sorts of circumstances. And one step further, therefore, each and every living thing is genetically unique and special. So the last thing that we want to be able to do is use the code originally found in DNA to get to the finished product of the proteins. The way we build the proteins, remember, are specific amino acids. So from DNA, we build RNA. The RNA has triplets called codons. The codons give us the specific amino acid sequence. So if we use these bases, AGG, CUC, etc., if we use these bases in a specific tool that we have called an amino acid chart, we'll be able to list the amino acids in order. To figure out the amino acid sequence, you need to remember to use the mRNA codons. If you use the mRNA codons for these charts, you'll get the correct order of amino acids to be able to identify the amino acids. Now in the last slide, we'll just do a couple of these. In the last slide, the first codon was AGG, and the second one was CUC. So if we use the wheel first, AGG, you start in the middle, go to the second ring, and then the outer ring. So A, and then you will stay in just this quadrant. A, G, to the outer one is G. You have that arginine. For the second amino acid, use the second codon, CUC. So you'd start C, go out to the U, and C. This particular one is called leucine. And as you see by this wheel, you notice that there are multiple codons that will give you the same amino acid. In other words, to get leucine, there's different possibilities. Using those RNA codons in this type of chart, the first one, AGG, use the first letter in this column, and it's A. The second letter is G. And the third letter, I'm going to come down here because we're only using these ones that are straight across from A. A, G, G. And again, I get arginine. For the second code, or for the second letter, or codon, I'm going to use CUC. C, U, and down to C. Which gives me leucine. So practice those, ask questions. I'm only giving you a couple examples in the screencast. They can be quite confusing, but once you have them down, lickety-split. You would never need to memorize any of these sequences. You use these two tools, which do the same thing. You use those to determine the amino acids. You would never need to memorize any of the amino acids or any of these sequences.